Superficial detoxification.
The current environmental conditions in our world subject human beings to a substantial and increasingly higher exposure to toxins. Such exposure begins with the air we breathe, the manipulated and denatured foods we eat, and the inadequate working environments. Inadequate lifestyles full of toxic habits, voluntary use of social toxins, and the ever-increasing psychic overload of stress and anxiety result in saturation of the biological regulation and control mechanisms of the body, paving the way for chronic disease.
With development of the homotoxicology theory, Hans Heinrich Reckeweg laid down the foundations for understanding the problem, but also provided us with the elements that will be essential for the process to treat homotoxicity. The mainstay of anti-homotoxic therapy is detoxification, essentially consisting of two parts, one aimed at patient education, and the other consisting of a step-by-step therapy that will depend on the circumstances of each individual patient. For this purpose, we start with the so-called superficial detoxification, aimed at working on the humoral phases in the disease evolution table, and which will allow us for inducing excretion mechanisms to remove homotoxins. Three remedies of anti-homotoxic therapy are basic and essential for superficial detoxification, namely Nux vomica homochord, berberis homochord, and lymphomyosote. Knowledge of the pharmacological of these remedies will allow for understanding their pharmacodynamics and how they act.
Nux vomica homochord. This